Good morning student. Today we are going to discuss some other effect of miRNA and P-bodies, processing bodies. Now stimulation of translation by miRNA. So far we have discussed what is miRNA, how miRNA actually processed, and what are the functions of miRNA. While discussing miRNA's function, we have must informed you that miRNA repress translation particularly for animal cells. But there are some other observation also there. So that's what one is stimulation of translation by miRNAs. MicroRNA can activate as well as repress translation. So the repression we have discussed how it paired with the 3' untranslated region and repress the translation. Here, Seitz and colleagues discovered positive action by miRNAs. They found that ARE, that means AU-rich element of human tumor necrosis factor alpha mRNA activates translation during serum starvation, which arrests the cell cycle in G1 phase. If you remember, serum starvation is the way generally in vitro we do cell cycle arrest. We try to maintain the cell in the G1, G0 phase. So serum starvation meaning we are not giving any serum, and as you know the serum contains many growth factors. So as there is lack of other types of growth factors, so naturally cells generally go for G1 phase, G0 G1 phase. So but at the time they have found that TNF alpha mRNA activates translation. So definitely starvation has an effect of this activation of translation process. In particular, miRNA 3693, this is a type of miRNA with the help of AGRO2, you know the protein AGRO1, and FXR1, another protein, activates translation of the TNF alpha mRNA in serum starved cell. MIR 3693 with the help of AGO2 can repress translation of mRNA in synchronized cells growing in serum. So see the difference here, serum plays a crucial role. When the serum is there, that time this particular miRNA with the help of the same agronaut proteins repress the translation, but when the starvation is going on, some other mechanisms definitely is playing and stimulate the translation of TNF alpha mRNA. Now translation repression and mRNA degradation and P-bodies. Let us see what is called P-bodies. These are the distinct foci formed by phase separation within the cytoplasm of eukaryotic cells, which consisting of many enzymes involved in mRNA turnover, meaning its degradation. P-bodies are highly conserved structure and have been observed in somatic cells originating from vertebrates and invertebrates. It also present in plant and yeast. To date, P-bodies have been demonstrated to play fundamental roles in general mRNA decay. What we know the mRNA decay, the normal process. Apart from that, nonsense mediated mRNA decay, AU-rich element mediated mRNA decay, and microRNA induced mRNA silencing. So processing bodies are the discrete collection of RNAs and proteins that are involved in mRNA decay and translation repression. These cellular foci are enriched in enzyme, as I already mentioned, that deadenylate mRNA, decap mRNA, and catalyze 5 to 3' degradation of mRNA. So these are the process of mRNA degradation, deadenylating, decapping. Okay, degradation of mRNA occurs by a non-RNAi-like mechanism. So in P-bodies, most likely the degradations are, as I mentioned, normal mRNA decay, nonsense mediated mRNA decay, AU-rich element decay, and some cases miRNA induced silencing also takes place. Now, translation, repression, mRNA degradation, and P-bodies. P-bodies are cellular foci where mRNA are destroyed or translationally repressed. One such type of function we have found in GW182. What is this? These family proteins are essential for miRNA mediated gene silencing in animal cells. They are recruited to mi targets through direct interaction with Argonauts proteins and promote target silencing. They do so by repressing translation and enhancing mRNA turnover. So GW182 is a family proteins, okay, is an essential part of which is actually discovered in Drosophila miRNA silencing mechanism, and that mechanism which was going on inside the P bodies. AGO1 probably recruits GW182, so it's a family of protein, to an mRNA within a P body that marks the mRNA for silencing. GW182 and AGO1 mediated RNA decay in P bodies appears to involve both deadenylation and decapping, followed by mRNA degradation by 5' 3' exonuclease. Now, there are some certain other observation also. Relief of repression in P bodies. Although many mRNAs are degraded in P bodies, many other are held and repressed there. In a liver cell, these are just observation. I am just giving some of the information. In a liver cell line, that's liver cell line HUH7, translation of CAT1 mRNA is repressed by miRNA. What is the miRNA? miR122, and the mRNA is sequestered in P bodies. Upon starvation, the translation repression of CAT1 mRNA is relieved and the mi migrates from P bodies to polysomes. So, starvations, these are all in vitro observation. So when you starve the cell, then there is no repression. That miRNA only migrates from the P bodies. This depression and translocation of mRNA depends on mRNA binding protein HUR. This is upstream region binding protein on its binding site at 3' untranslated region of the mRNA. UR meaning untranslated region binding protein. Now, apart from, we need to know some of the other small RNAs. We already know siRNA, miRNA, and there is piRNA. I have discussed the subunits of Argonauts protein. There is a piWI subunit. There are some RNA scientists have discovered that they mention piRNA. Other small RNAs have been found, although the function of these RNAs are largely still unknown. One such example is the endo-siRNA. We have studied siRNA, but there is a specific name we are mentioning, endo-siRNAs of Drosophila, which are made from Drosophila genes as double-stranded RNA precursors. These RNAs, meaning this is endo-siRNA, which identified in Drosophila, these RNAs blurred the line between si and miRNAs. They may protect somatic cell from transposons effect. So these are the some of the information regarding other functions of mi and the P bodies and some other small RNAs and their function. Thank you very much.